Hello, biology students. Today I want to talk to you a little bit about some background ideas in chemistry so you can better understand what enzymes do. Have you ever wondered why a pile of paper, such as in this very old book on moths, this book has been around for 100 years, and yet if I were to put a match to it, it would disappear relatively quickly. So how can we explain that this book is stable for 100 years and then at the same time I could put a match to it and it would disappear in an instant in flame and release all sorts of energy? The other thing I want to say about this book, it's 100 years old and it is beginning to deteriorate. So what's up with that? How do we explain the deterioration of the book, even though this is a brand new copy that I purchased? Let's talk about some background chemistry ideas so you can understand those ideas. First thing, let's remind ourselves what a chemical reaction is. It's the breaking and reforming of bonds between atoms and molecules. So, for example, here's a famous chemical equation. 6 waters and 6 carbon dioxides form 1 molecule of sugar, or glucose, and 6 molecules of oxygen. You'll probably recognize this as the famous photosynthesis reaction. What I want to point out here is that in order to go from water and carbon dioxide, old bonds have to be broken down and new bonds have to be formed to make the sugar molecule and the oxygen molecule. Virtually all chemical reactions require some energy input to get the reaction going. Let's say that again. All chemical reactions require some energy input to get the reaction to occur. And this is called activation energy. The activation energy we can depict like this. Here we have a graph and there's energy on the y-axis, and this is the time course of the reaction. And in this case, the reactants require an input of energy in order for them to form products down here. And we can notice that the reactants have an energy level that they're at, and the products an energy level they're at. In this case, the reactants contain more energy in them than the products do. But this EA here is the activation energy. It's the amount of energy that needed to be input. And in this case, energy is going to be given off in the long run because these products don't store as much energy. And over here, we have reactants with a large activation energy, and here that's denoted by A, and the products formed, and they contain more energy than the reactants did. And so in this case, the products store the energy, and energy is not released, but it is absorbed into the products. And this is exactly what happens in photosynthesis, where the sugar stores the energy. And this is what happens in cellular respiration. So we can think of activation energy as a barrier, right? It is what prevents a chemical reaction from happening spontaneously and makes it only occur at a very slow rate. To get back to the question of why doesn't paper burn spontaneously? Because it requires the energy input supplied by a match to get it going. And then once it gets going, the energy produced by the chemical reaction is enough to keep it self-sustaining. You don't have to keep putting any more energy in because the reaction of the paper and the oxygen produces so much energy that it continues to burn. So what enzymes do is they lower the activation energy barrier. And they do this at the low temperature that's found inside cells, so you don't need to put in additional energy. They don't affect the ratio of energy in the reactants and the products, but all they do is hasten reactions that would occur eventually. And that answers our question about why this book is deteriorating. Because even though a match would cause it to deteriorate very quickly, still oxygen reacts very slowly with the paper in this book. And so over time, we could say the book does a slow burn and the paper deteriorates by the same chemical reaction that happens in burning, but it just goes really slowly. Now, enzymes lower the activation barrier, and they do this in a number of ways. They orient the substrates correctly towards each other. They can put a strain on the bonds in the substrate. They can provide a favorable microenvironment by changing the pH. They can briefly bond to the substrate and then twist it or apply forces. And so what we see is we get a graph like this, that here is the reaction without an enzyme, and here is the reaction with it. And you'll notice that the activation energy barrier has been brought down lower in this case, about half, and in many cases much, much lower. So the enzyme carries out the reaction very, very quickly. Now, we said that the chemical reactions will occur slowly without the use of an enzyme, and that's because our view of chemical reactions is they occur by collisions between molecules. And the collisions have to have enough energy, enough kinetic energy when they smack into each other, and the molecules must have to hit each other at just the right angle in order to get the reaction to occur. So at the ambient temperature of a cell, these collisions that have enough energy and that are oriented in just the right way happen very, very rarely. But what an enzyme does is make them happen much more frequently, and therefore products are formed at a very high rate. We remember that the enzyme has an active site, and the active site is where the substrate can fit. And now the enzyme has the substrate just where it wants it and can provide just the right orientation so the collisions can happen in just the right angle, and it will cause the products to be formed. And so what we see is the substrates fit into the enzyme, create an enzyme substrate complex. Then the enzyme does its magic, converts them into a product, and then it releases the products, and now the enzyme is back and ready to catalyze another reaction. In order to be called a catalyst, the enzyme has to meet two criteria. One is it has to speed up the rate of a reaction compared to what would occur at normal ambient conditions. That means the conditions in the cell or whatever the environment has in that place. And two, it has to itself be unchanged by the chemical reaction and be ready to participate in another one after it's done its job. So I hope you understand after the end of this talk what activation energy is, what an enzyme does to the activation energy, and that you understand that enzymes don't create any chemical reactions that wouldn't occur in nature, but they just speed them up, often millions of times faster. Let me know if you have any questions by putting your questions into the shared Google Doc of questions. I'll talk to you soon.